Hey everyone, in this lesson we are going to talk about glycogenolysis, and I request you to please watch my previous video on glycogenesis before you watch this video. So glycogenolysis, basically if you break down the word, first is glycogen and lysis. Lysis means breakdown, therefore glycogenolysis means breakdown or degradation of glycogen. So glycogen is predominantly synthesized in liver and skeletal muscle. So the main purpose of glycogenolysis is to mobilize the stored glycogen from liver and skeletal muscle to provide immediate energy to skeletal muscle during extensive exercise or to maintain blood glucose level during fasting. Now, glycogenolysis generates glucose in the form of glucose 1-phosphate and free glucose, which are produced in the ratio of 10:1. Basically, what it means that every 10 glucose 1-phosphate molecule, there is one free glucose is released during the process of glycogenolysis. So the one very important step in glycogenolysis is the cleavage of an alpha 1,4 glycosidic bond. So suppose here we have the glycogen molecule. Basically glycogen molecule is a multi-branched polysaccharide of glucose monomers. So these are multi-branched and glucose molecules, because all these green dots, they are glucose monomers. So here if we look at closely, this is the glucose monomers, and these glucose monomers are linked to each other via alpha 1,4 glycosidic bonds. And this one terminal glucose molecule, which is in red, that is called as non-reducing end. Basically non-reducing ends are identified as the terminal glucose molecule with free hydroxyl group on its on its carbon 4 position. So this terminal glucose molecule is identified as non-reducing end. Now, before we talk about anything, there is one important thing you need to remember, that glycogen chains are extended or degraded from their non-reducing end. Basically, during glycogen synthesis, glucose molecules are added from the non-reducing end, and during glycogenolysis, the glucose molecules are broken down from the non-reducing end. So this is something very important to remember, that either extension or degradation, that has to happen from the non-reducing end of the glycogen chain. Now let's talk about the cleavage of an alpha 1,4 glycosidic bond. So this cleavage is catalyzed by an enzyme called glycogen phosphorylase enzyme. So basically this is the rate limiting enzyme which catalyzes the cleavage of glucose residues from the non-reducing end of the glycogen chain. So by doing that, it releases this glucose molecule in the form of glucose 1-phosphate. So this is a phosphorylosis reaction. So in phosphorylosis reaction, there is a transfer of inorganic phosphate onto the hydroxyl group of carbon 1 position of terminal glucose molecule. So as soon as the inorganic phosphate group is transferred onto the hydroxyl group of carbon 1 position of terminal glucose molecule, it initiates the breakdown of alpha 1 glycosidic bond, and which releases this terminal glucose molecule in the form of glucose 1-phosphate. So this is how glucose 1-phosphate molecule is generated by breaking alpha 1 glycosidic bond with the help of enzyme glycogen phosphorylase. And this is the remaining glycogen chain. So subsequently glycogen phosphorylase enzyme is going to break down this alpha 1 glycosidic bond, and again releasing this glucose molecule in the form of glucose 1-phosphate. So the cycle continues with the help of glycogen phosphorylase enzyme. So this is the important reaction in glycogenolysis, which involves the cleavage of an alpha 1,4 glycosidic bond by glycogen phosphorylase enzyme. So now let's talk about the steps involved in glycogenolysis. So the first step in glycogenolysis is the shortening of chain. So here we have this glycogen molecule and the glucose molecule, which are circled with the black line. Those are the non-reducing ends, and here this glucose molecule of this branch is connected to the another glucose molecule of the main glycogen chain by alpha 1,6 glycosidic bond, and which is also called as branch point. So glycogen phosphorylase enzyme play an important role in shortening of glycogen chain. Basically, the enzyme releases glucose molecules one by one from their non-reducing end until there are only 4 glucose molecules left on the chain. For instance, I have placed this red line. So the any glucose molecule before this red line, they are all removed one by one with the help of glycogen phosphorylase enzyme by breaking alpha 1,4 glycosidic bond. And all the 4 glucose molecules after the red line, all these 4 glucose molecule, they are retained on the glycogen chain. So this is the resulting structure where you can see that there are only 4 glucose molecules left on the chain, and these are the branches where the where the glucose molecule with the red circle that is attached to another glucose molecule at the branching point, at branch point by alpha 1,6 glycosidic bond. And also remember that all these glucose molecules which were released by glycogen phosphorylase enzyme, those are released in the form of glucose 1 phosphate when inorganic phosphate molecule is transferred to the hydroxyl group of terminal glucose molecule on carbon 1 position. Also, the resulting structure is called limit dextrin. So the next step in glycogenolysis is the removal of branches. So this is the resulting structure from the previous step of shortening of glycogen chain by glycogen phosphorylase enzyme. So in this step, glycogen residues which are located near the branches, so these are the glycogen residues which are located near the branch, these are removed in a two-step process which requires a bifunctional debranching enzyme. Now this enzyme is called debranching because we are removing the branches. If we were to synthesize the branch, which we already saw in glycogenesis process, the enzyme is called as branching enzyme. So this is not so difficult. This is very easy, that to synthesize branch, the enzyme is called branching, and to remove the branch, the enzyme is called debranching enzyme. So as I mentioned, this is a two-step reaction. So in the first step, the transferase activity of the enzyme shifts outer 3 glucose residues from the branch to a nearby non-reducing end. So suppose here we have 3 outer glucose molecule which are shifted to the nearby non-reducing end. So this is the resulting molecule where you can see that three glucose molecules, these are moved to the closed or the nearby non-reducing end. So basically, debranching enzyme, what it does is it actually breaks the alpha 1,4 glycosidic bond between these 2 glucose molecules and take this glucose molecule and reattaches this glucose molecule to the non-reducing end by forming alpha 1 glycosidic bond. So in the process, the alpha 1 glycosidic bond is broken and alpha 1 glycosidic bond is also formed to reattaches these glucose molecules to the non-reducing end, and therefore the enzyme is called to have 4,4 transferase activity. Now the next step is the removal of this single glucose residue at the branch point where this glucose molecule is connected to another glucose molecule by alpha 1,6 glycosidic bond. So this single glucose residue is then hydrolytically removed as a free glucose with the help of 1,6 glycosidase activity of debranching enzyme. So basically debranching enzyme, as I mentioned, it is a bifunctional enzyme, and now you know that why it is bifunctional, because it has two different types of activity. It has transferase activity and it has glucosidase activity. And here you can see that the single glucose residues are hydrolyzed and they are released as a free glucose, and this is the resulting structure. So once the branches are transferred and the glucosyl or the glucose molecules at C6 position is hydrolyzed, the remaining glycogen chain can then be shortened with the help of glycogen phosphorylase enzyme by breaking down this alpha 1,4 glycosidic bond. So the cycle of shortening chain and removal of branching continues until the glycogen molecule is completely degraded. For instance, this is our resulting structure, and here this is the glycogen, this is the branch, and this is the branch point where two glucose molecules are connected to each other by alpha 1,6 glycosidic bond. So the first step is the shortening of chain, and which is catalyzed by the enzyme glycogen phosphorylase, which basically releases the uh, glucose residues from the non-reducing end of the glycogen chain by cleaving the alpha 1 for glycosidic bond, and releases glucose molecules one by one in the form of glucose 1 phosphate until there are only 4 glucose residues left on the glycogen chain. So this is the resulting structure, and the next step is the removal of branches. So this is the branch, and the reaction is catalyzed by debranching enzyme, which basically shifts the 3 outermost glucose residues to the nearby non-reducing end of the glycogen chain, and this is performed by this reaction is catalyzed by transferase activity, and the resulting molecule, where you can see that the 3 outermost 3 glucose residues are then transferred to the non-reducing end by forming the alpha 1 4 glycosidic bond. And next step would be to remove or release this single glucose residues which is left on the glycogen chain, and that is catalyzed by glucosidase activity of debranching enzyme, and the enzyme basically hydrolyze this single glucose residue and release this glucose residue in the form of free glucose, and the resulting structure is one single glycogen chain. So the cycle continues, as I mentioned, the cycle continues until the glycogen is completely degraded or broken down. So one thing to remember from this process, that glycogenolysis generates glucose 1 phosphate and free glucose molecules. So now you know that there are more glucose 1 phosphate molecules are generated than releasing the free glucose. So next thing I want to talk about, because as I mentioned in my previous video, that glycogen is synthesized predominantly in liver and skeletal muscle. That means it is also broken down predominantly in liver and skeletal muscle. So I just want to quickly talk about what happens to glucose 1 phosphate once the glycogen is broken down in liver and skeletal muscle. So here we have liver and skeletal muscle, and this is the glycogen in both liver and skeletal muscle, which is then broken down with subsequent steps. It is broken down into glucose 1 phosphate. So in liver, glucose 1 phosphate is isomerized into glucose 6 phosphate with the help of phosphoglucomutase, and this is a reversible reaction. And it is no different in skeletal muscle, where glucose 1 phosphate is isomerized into glucose 6 phosphate with the help of phosphoglucomutase enzyme. Now in liver, once the glucose 6 phosphate is synthesized, it is then converted into glucose with the help of glucose 6 phosphatase enzyme. And once this glucose is synthesized, it is then enters the glucose which is synthesized here or generated here, it then enters into the bloodstream to maintain blood glucose during fasting. So this is the main purpose in liver, that once the glycogen is broken down into glucose 1 phosphate with subsequent reaction, it will be converted into glucose, and glucose is then will be sent into the blood circulation to maintain the blood glucose level during fasting. Now it is a little bit different in skeletal muscle. Once this glucose 6 phosphate is synthesized, it actually enters into glycolysis pathway to synthesize pyruvate, and pyruvate is then subsequently enters into the TCA cycle to synthesize energy in the form of ATP. And there is a reason behind that. skeletal muscle actually lacks this glucose 6 phosphatase enzyme, and because it does not have this glucose 6 phosphatase enzyme, glucose 6 phosphate cannot be converted into glucose. Therefore, glucose 6 phosphate directly enters into glycolysis to synthesize energy in the form of ATP. So this is very important because if we are doing some exercise, or suppose in athletes who are constantly undergoing extensive training, skeletal muscle actually breaks down glycogen and releases energy in the form of ATP. So it provides immediate energy during extensive exercise. So this is the most important function, in liver and skeletal muscle. In liver, it is basically the main purpose of glycogenolysis is to maintain the blood glucose during fasting, and in skeletal muscle, glycogenolysis provides immediate energy in the form of ATP during extensive exercise. So now I just want to quickly go through the process of, review the steps of both glycogenesis and glycogenolysis, so you can actually compare both the process synthesis and breakdown of glycogen. So in glycogenesis, glucose is converted into glucose 6 phosphate with the help of enzyme glucokinase and hexokinase, which is also an ATP dependent process. Subsequently, glucose 6 phosphate is then converted into glucose 1 phosphate with the help of enzyme phosphoglucomutase, and glucose 1 phosphate is then converted into UDP glucose with the help of enzyme UDP glucose pyrophosphorylase, which also requires UTP. And in the next step, glycogen synthase enzyme uses this glycogen primer and extend this glycogen primer, or extend this glycogen short glycogen chain by adding glucose molecule from the non-reducing end, and that glucose molecule is donated by UDP glucose. And most important function of glycogen synthase is to form this alpha 1,4 glycosidic bond, and in the process UDP is released, and subsequently as the more glucose molecules are added, the glycogen chain is also then being extended, and next step is to form the branches. So as I mentioned, to form the branches, the enzyme responsible for forming the branches is called glycogen branching enzyme, which basically form the branches between two glucose molecules which are connected to each other by alpha 1,6 glycosidic bond. So this is the process, or these are the steps involved in glycogenesis. Now let's talk about glycogenolysis. So this is the glycogen chain, and this is the branches where two glucose molecules are attached by alpha 1,6 glycosidic bond. So the first step in glycogenolysis is the shortening of chain, which is catalyzed by glycogen phosphorylase enzyme, which basically releases glucose molecule from the non-reducing end of the glycogen chain by cleaving the alpha 1,4 glycosidic bond. So these glucose molecules are released until there are only 4 glucose residues left on the chain. So the next step, also the one thing to remember is glucose molecules are released in the form of glucose 1 phosphate molecules. Now next step is the removal of branches, which is catalyzed by the enzyme debranching enzyme, that is the transferase activity of debranching enzyme, which basically shifts this outer 3 glucose molecules to the nearest non-reducing end of the glycogen chain. So here you can see that those 3 glucose molecules are moved here by forming the alpha 1,4 glycosidic bond. And in the next step is the removal of the single glucose residue. So this single glucose residue is hydrolyzed by glucosidase activity of debranching enzyme, which releases this glucose as a free glucose molecule, and now we have the single glycogen chain. So these steps keep on repeating until the glycogen chain is completely degraded or broken down. So lastly, I quickly want to talk about the clinical relevance of this glycogenolysis pathway. So before I mention anything, I quickly want to mention that there is also a small amount of 1 to 3% glycogen which is continuously degraded by the enzyme which is located in lysosome, which is called as alpha 1,4 glycosidase, also known as acid maltase enzyme, which degrade glycogen and releases glucose. And when there is a deficiency of this enzyme, it causes an accumulation of glycogen in vacuoles inside the lysosome, which resulting in the serious glycogen storage disease type 2, which is also called as Pompe disease. Now in the second condition, which is known as, suppose if the glycogen phosphorylase enzyme which is deficient in muscle, it causes glycogen storage disease 5, which is also called as McArdle disease. And if the enzyme is deficient in liver, it causes glycogen storage disease 6, which is also called as Her's disease. So in liver, when this enzyme is deficient, it causes an enlargement of liver, and it may or may not causes hypoglycemia in the patients who are suffering from this disease. Whereas in the muscle, when this enzyme is deficient, it causes muscle cramping in patients who are suffering from this disease. And unlike liver, deficiency of this enzyme does not cause any structural abnormality in muscle. So now the second condition is Cori disease, which is also glycogen storage disease 3. In this condition, there is a deficiency of debranching enzyme, both transferase activity and glucosidase activity. And patients who are suffering from this disease, the deficiency of the enzyme can cause fasting hypoglycemia in these patients. And glycogen structure in these patients are abnormal because it only has 4 or 1 glucosyl residue at the branch point, because of the debranching and because of the deficiency of debranching enzyme, it is not able to remove either these 3 glucose molecules or this one single glucose molecules. So that means there is a structural abnormality of glycogen itself in these patients who are suffering from this disease. So I really hope that you learned something new from this video, and I hope that this lesson is helpful to you. And if you do so, please like and share the video and subscribe the channel, and I will see you soon next time. Thank you so much.